Momita, please.
Okay, sir. Then I'm presenting.
You tell me when it is full.
Is it visible, sir?
Yes, it is coming.
1 minute.
I'm
Yeah,
making
it's
it full
coming.
screen.
It's coming. Okay, good afternoon, everyone. Today we have with us Dr. Chandrama Mukherjee, Assistant Professor and Ramalingaswamy Fellow. She is from RNA Biology Laboratory of Department of Life Sciences and School of Biotechnology, Presidency University, Kolkata. Thank you very much, Chandrama, for accepting my invitation readily, even though you have a very busy schedule recently. So we are honored and overwhelmed. Her research area encompasses various aspects of RNA biology, parasitology, biochemistry, and molecular biology. Recently, she has taken interest to work on aquatic ecology in collaboration with Marine Ecology Laboratory. She has obtained her doctorate from Bose Institute and Jadavpur University in 2008 on functional genomics of cell cycle control genes in entamoeba histology. Subsequently, she did postdoctoral research at Department of Cellular and Molecular Biochemistry at Ohio State University, Columbus, USA. She was also a research associate in the same institute. She was awarded the postdoctoral fellowship from American Heart Association. She was recipient of Research Associate Fellowship from CSIR, Government of India. She was awarded with prestigious Ramalingaswamy Re-Entry Fellowship from DBT. She has published more than 13 research publications in highly reputed journals, having phenomenal 1,568 citations. Furthermore, one of her publications in Cell Reports has a 64 citations. She has been serving as reviewer for many national and international journals, as well as various projects. With this introduction, I am requesting Dr. Mukherjee to deliver her talk titled Integrating Molecular Tools to Study Aquatic Ecosystem. Over to you, Dr.
Thank you, Shumi. Thank you for such a nice introduction. I'm really overwhelmed to get this invitation from the Aquaphile Mail group. It's really nice, and I appreciate the effort by your team. So, and also I'd like to thank all the audience who are here for a scientific session on a Sunday afternoon. Though in this COVID situation, now the days are really mixed up. So every day is Sunday and every day is weekday. So I'm going to present my talk. Just give me 1 minute. Yeah, it will take some time. Can you see the screen?
No, Chandrama. Yes, it has come. It has come. Please make it full screen.
Yeah.
It's perfect.
So now is it visible?
Yes, perfect.
Okay, thank you. So the title of my talk is Integrating Molecular Tools to Study Aquatic Ecosystem. As you heard from the introduction, by training, I am a molecular and cell biologist. I have never worked with any kind of ecological system, but this is due to my colleague, Dr. Shumit Mondal, who actually excites me about this aquatic ecosystem. So this ecosystem is like the 70% of the Earth's surface is covered with water, and within this aquatic environment lives a whole myriad of aquatic organisms. These life forms are vital to the global ecosystem and sustainability. And as we know, and there are like all different speeches from yesterday's, the terrific talks by the different speakers, you all know like how the ecosystem is, aquatic ecosystem is like maintaining its dynamicity. So I'm not going to detail, I'm not the person to talk about those things. But what I'd like to say that this rapid growth of human populations and development, in particular, land reclamation of coastal regions, harvesting of fishery resources, and aquaculture practices has caused much public concern about their negative impacts on the environment. And our interest in understanding the function of marine ecosystems has been accelerated in recent years with growing recognition of their importance in human life. So, marine biology is on years with growing recognition of their importance in human life. And this is a really an excellent system to study. It is an interdisciplinary study of the life in the oceans and inland seas of the world. And this subject is on the threshold of an exciting new frontier of scientific discoveries and economic growth. And biochemistry. So marine biology intersects the field of biological oceanography, physiology, cell biology, biochemistry, and microbiology in association with chemistry, because all are related and we need all of these subjects to better understand this aquatic ecosystem. The progress in the research of biological sciences happened during the past 2 decades with the development of sophisticated technologies and instruments. It has the potential of offering new capabilities to tackle problems associated with global climate change, biodiversity, environment quality, and use of marine living resources. So, if we club the different branches of molecular biology and we want to understand the marine biology, that generates a new field which we call as marine molecular biology. So you can see here with the help of these different tools and also with the old traditional, the taxonomic method, we can characterize the new species, or we can actually determine the relation of the species with the habitat, which is very important to understand the biodiversity. So marine biologists study oceanic life in relation to marine environment that often range from small to global scale, whereas the molecular biologists study biological events in terms of the physiochemical properties of molecules. The immediate benefits of the collaborative research between these two disciplines could include improved management of fisheries resources, enhanced production of useful species through aquaculture isolation and development of natural products, better information about environmental degradation. So I would like to give you some examples of how this integration of molecular tools help us to better understand the ecological problems. For example, gene encoding a specific photopigment, rhodopsin, was discovered within a large fragment of DNA belonging to an as yet uncultured bacterium that is ubiquitous in the ocean. Recombinant expression and biochemical characterization showed that this is a new type of light-driven energy generation in the sea, which researchers never suspected before. The abundant pelagic bacteria that harbored this gene may be a new types of phototrophs and that may alter the current perspective of complex energy flow systems in the sea. This discovery is an excellent example of how molecular biological approach in the field of marine biology can bring new insight to our vision of the function of marine ecosystems. So, with the help of molecular biology techniques in association with bioinformatics open the new field in this marine molecular biology. So what we do, what are the approaches we took? So the first and foremost which comes into the picture is the genomics or genome sequencing. So in genomics, two approaches have been used. The number one is that where the whole genome sequencing of key organisms have performed, and the other one is the analysis of partial or whole genome sequences from environmental samples. Sequencing of total genomes of many organisms is a rapidly advancing research area. In a major scientific breakthrough, researchers have decoded the first complete set of genetic instructions from a microorganism that confirms the existence of the third major branch of life on Earth. Researchers have sequenced the genetic information or genome of Methanococcus genusci, a single-cell microorganism known as an archaea. The microbe is a methane-producing thermophile which is discovered at the base of a Pacific Ocean thermal vent. And this thermal vent means like where the temperature of water is near 100 degrees. So this organism physiologically specialized to undergo all the metabolic reactions at that high temperature. It is strictly anaerobic and uses carbon dioxide as its sole carbon source. The discovery provides fundamental information about life on Earth, its evolution and diversity. And it has been known that 52% of the proteins of this particular organism have been assigned to some functions. So the remaining percentage of the protein is still unknown, and we don't know how the proteins of this particular organism like greatly exhibited that hyperthermophilic properties so that it can adapt to that extreme environments. And not only that, identification of this particular organism also helps the identification of novel cofactors which are recently utilized by various biotechnological applications. We can take another example like where the complete genome of Pelagibacter ubique was published in 2005. So this particular organism, this belongs to Sartuclade and which is actually identified in a Saragossa assay near the Bermuda Triangle. And this particular organism is the most successful free-living organism on the planet, accounting for around 25% of all microbial life. Unlike most other marine heterotrophs, it has a small genome. In fact, the smallest genome of any free-living cell in nature. And if you just see about its genetic composition, you will wonder to see that it is such a small genome, but it has the complete biosynthetic pathways for all 20 amino acids and also some few cofactors. And interestingly to note, like this particular organism does not contain any pseudogenes, introns, transposons, or any other kind of extra chromosomal elements or intents. So this organism actually has a capability to avoid the mutations. So scientists collected this organism throughout the world, and it was very surprising to notice that this particular organism collected from whatever independent of its geographical locations, it has no mutation. So, and this is really important. This particular organism is really important for the growth of this ocean alate. Excuse me. Sorry, just. Sorry. So okay. Sorry guys for the disturbance. Okay, so next, after the genomics, so genomics, what we have discussed about the where we are actually determining the complete genome set. And also the second approach is also very crucial in the study of marine life, where the environmental samples could be processed to analyze complete or partial genome sequence of marine organisms present in the sample. So this is known as metagenomics. So where you can isolate a mixture of the organisms from the natural resources, either it could be soil or water or any sources from the environment, and we can get a mixture of organisms, and then we can directly sequencing to get a mixture of all the different organisms present in that mixture. So This approach provides novel insights into the ecology, evolution, and metabolism of uncultured microorganisms in nature. So in the past, there is some great association between among the different ecologists and microbiologists and molecular biologists who have developed MetaLook software for visualization, analysis, and comparison of marine ecological, genomic, and metagenomic data with respect to habitat parameters. So if you just look at the map, you will see, look at my cursor. So this is the world map, and there are some places. So if you just go to this website, and if you just go to these particular places, you will see the genetic diversity, you will see all the species which are collected or identified there. And so this provides a great resources for all the ecologists, or the specifically the marine ecologists, to analyze the different species in the different geographical locations. So how the metagenomics analysis has been performed? So as I said initially, like the samples are collected from the different resources. So all these are environmental samples. It's a mixed culture. There is no such pure strain is present here. So from these environmental samples, if you just like isolate the DNA, and then you can do a DNA sequence analysis, either you can do a whole genome sequencing, or you can just go for a partial PCR amplification, then you can make a metagenomic library out of this particular complex mixture. And then this library either can be hybridized for the functional screening to identify which type of enzymes activities are present in the mixture, or you can do a homology screening to identify which homologous genes are present in that complex mixture. And obviously, it involves a lot of bioinformatics to provide us the hits, and then whenever we get the candidates or here labeled as hits, we can sequence, then expression and functional analysis can be performed. So this is a very powerful tool to give us the idea of the different organisms present in a particular sample. So using this technique, there are a lot of different novel enzymes have been identified. And you can see from this table, like using this metagenomic approach, the different ocean sampling project have identified more than 100 enzymes from the deep sea microbes. So recent advances in molecular techniques are adequate to describe the microbial diversity in a marine sample based on 16S ribosomal RNA sequence diversity. This approach has dramatically changed our perspective in the area of microbial diversity, leading to the development of a 16S rRNA gene, which is highly conserved in bacteria. So this particular gene has been selected as a marker gene for phylogenetic analysis. And taking advantage of the variation within 16S rRNA gene sequences of different bacteria species has driven a revolution in our knowledge in bacterial ecology over the last decade. So how you can do that? Suppose like if you can see there is a sponge and you can do the DNA extraction, we can do the PCR amplification, and there are some kind of like universal primer sets are already obtained. So with that universal primer sets, you can do the specifically amplify the 16S ribosomal gene from the organism, and then cloning into different vectors. From that we can sequence, and from the sequences what we obtained, we can definitely construct a phylogenetic tree with the help of some other kind of softwares, like they have been used. The tree puzzle is a computer program to reconstruct phylogenetic trees from molecular sequence data by maximum likelihood. So bacteria have been used, like for bacterial identification, this 16S rRNA technology has been applied. But for the fungal identification, that is based on the 18S rRNA sequence. So there actually the 18S rRNA and also the other internal transcribed sequence or ITS have been used for determining the sequence variation. So other techniques which are widely used are like denaturing gradient gel electrophoresis. So this technique is widely used to detect the phylogenetic fingerprint of diverse marine microorganisms. So from the marine microorganisms, the genomic DNA isolated, and then this isolated DNA are side by side analyzed by digestion with several restriction enzymes, and also PCR amplified using the 16 primers for the 16S ribosomal gene. So, and these products have been analyzed in agarose gel to check the band pattern. Now at the right panel, you are seeing a gel picture where these particular DNA products have been analyzed, and each band represents each different species. So now for further to know what is the identity of these bands, what usually have done that individual bands can be excised, re-amplified and sequenced to obtain phylogenetic information. Thus, environmental genomics is also revealing bacterial interactions with marine animals. A recent study discovered 430 novel bacterial types associated with 3 floral species. In this investigation, they actually have taken a genome shotgun sequence. What is genome shotgun technique? So what is this technique? In this technique, with the help of metagenomics, the genomes are cloned and then multiple genomes are shared into very smaller fragments of different size, and then this unordered and those smaller fragment of genomes are sequenced. Now, all the sequences what we obtain from this analysis are unordered, not like paired up. So then the different computational automated assembly have been used to make it in a like a properly overlapping sequence segments, and then we can actually determine from these overlapping sequence segments, we can actually determine the genomic sequences. So using this technique, using this technique, so 1,800 new uncultured marine prokaryotic species have been identified, which includes 148 previously unknown bacterial phylotypes, and that have been estimated from only 1,500 liters of oligotrophic seawater. So in this study, over 1.2 million previously unknown genes have been identified thanks to the power of genomics. And this includes actually covers many previously characterized genes along with many uncharacterized genes too. So this project demonstrated that shotgun sequencing provides a wealth of phylogenetic markers that can be used to assess the phylogenetic diversity of a sample with more power than conventional PCR-based rRNA studies. So metagenomics, or the culture-independent genomic analysis of an assemblage of microorganisms, has the potential to answer fundamental questions in microbial ecology. So this approach has offered very useful information at the genome level is the massive marine prokaryotic metagenome sequencing project, which is providing a tremendous database for discovering metabolic capabilities and new ways to conceptualize and study prokaryotic biodiversity. And not only that, it helps us to determine the bacterial interaction with the other marine animals and the overall the influence on animal populations and ecosystem functions. So the another expression or another technique which is widely used for determining the different bacteria or to basically determine the relationship of the microorganism with the marine microorganism with the environment is microarray. So in microarray, what we usually do, we measure the expression of the RNA. So suppose you have isolated microorganisms from two different conditions, then you can just extract the RNA from these two different samples, and you should have a reference from where you are isolating the RNA to. And then all this RNA sample, suppose in this figure as you've seen here, that one biological sample 1 and the another one the biological sample 2. So we are getting the samples from RNA sample 1 and another one RNA sample 2. And also we have the RNA extracted from our reference sample, RNA reference. So with all this RNA, the three different RNA samples, what we usually do, we make the cDNA, we did a fluorescent labeling, and then we do a hybridize this cDNA library with a microarray chip. So what is a microarray chip? That's a chip where we already have all the different available species which we know, the products or the genetic materials, the DNA probes from those known organisms that's already in the chip. So with the chip is now hybridized with these samples, and then the image acquisition analysis performed. And from the image acquisition analysis, the statistical analysis usually have been performed to determine the differential expression or the functional analysis or the pathway analysis. But remember, in this particular microarray experiment, we need to know we have to have that confidence that this microarray chip has a very enriched collection of all the different gene sets available in the microorganisms. And using this technique, recently there was a very big microarray have been done, which is named as the Micro-tools microarray, where the different marine microbial taxa have been analyzed. As you can see, like not only that, like it also helps to determine the phylogenetic tree from the different domains of life. So I just want to mention that this microarray is more powerful than the previous genomes because not it only determines the sequence, but also it will help you to understand the relation of that microorganism with its environment. And there are like different applications in the marine ecology. So there is one paper, a recent paper, where the phytoplankton transcriptomic and physiological analysis have been done in response to fixed nitrogen. And also there are several studies which want to address the effects of nutrient enrichment on surface microbial community. So as you can see, this is just the two examples. If you go to PubMed, you will see like there are like several reports where people have used the microarray techniques to determine the impact of that marine microorganism with its environment, or how the different physiological parameters have been all these like different parameters like changing the microorganisms to adapt with the environment. So there's like tons of literatures. But all techniques have its own advantages and disadvantages. So this microarray technique has a particular limitation. What is that? If you remember that I have told you, like the chip, this chip, it should have all the different combination of available gene information there. Suppose if a gene information is not there, then it will not pop up during the hybridization process. So what does that mean? That means, so the chip will have all the known organisms, all the genetic contents of the known organisms. But suppose you are working with an unknown organism where there is no genetic information available. So then how are you going to use a microarray to determine its relationship or its genetic diversity in the system? So then you cannot use the microarray technique because it is not known. We don't know the genetic composition of that particular microorganism. So there we can use the next generation sequencing. What is it? And it can be used for both for DNA and RNA. So you can make either a DNA library or a RNA library. And then for RNA library construction, you can simply use RT-PCR, make the cDNAs, and then there are like the commercially adapters are available. We can do adapter ligation, and after this adapter ligation, the PCRs are made, and then the library is being sent for sequencing. So as for RNA, the DNA library also can be made in a similar way, where the genomic DNA has to be extracted from the organism first, and then it has to be sheared or it has to be fragmented. And the PCR, you can use the PCRs to pick up the particular amplicons, and then do the same thing. The adapters are ligated, and then do the PCRs to make the sequencing library. And with the help of these sequencing libraries, you can sequence all different types of known and unknown organisms. So this actually is now widely used for all the different kind of expression studies, because people are now more interested to determine the relationship of a microorganism or marine microorganism with its habitat and how that influences the others which are present in the system. So now I'm going to talk about a very specific example, because before that, these all are the different tools what I want to share with you, what people have already used. So now why I am here, why I came into this particular ecological problem, because I think that this ecological problem is a very wonderful system to address with the help of the molecular biology. So Dr. Sumit Mondal has a long project on this particular gastropod, which is a Bellamia benjolensis, and this particular organism is like edible mollusca. It is serving as a very high rich protein food into many tribal areas in India. And there are also previous reports about this particular organism that it can be served as a good bioindicator species in biomonitoring studies. And not only that, this contributes significantly to the benthic biomass. So there are like changes in physiological responses of Bellamia benjolensis with respect to the predicted future thermal elevation, and that is the area of interest of my collaborator, Dr. Sumit Mondal. So his group has already published a paper where they have showed that how the increase in temperature is changing the physiological energetics of this particular mollusca. And they have have used the different assays which are usually used in any standard ecological lab to determine the physiological response with respect to the environment. So Sritama and Shanton, they showed that when you increase the temperature, there is a change in the total weight of the organism, and they have done the study at different temperatures, 25, 30, and 35 degree. So if you see in the right panel, you can see that with increase in the temperature, there is a overall decrease in the total weight. So I'm just for simplicity, I just wanted to show you a simple picture, but that study is beautiful, and they have done many experiments to prove that all other different physiological parameters like ingestion rate, excretion rate, the growth mass, everything is affected when there is a change in the temperature or when there is an elevation in the temperature. So now when Sumit actually came to me and shared his wonderful results, I was really interested, and I thought from a molecular biology angle that how this particular organism responds to heat stress, and what are the molecular players in regulating this phenomenon. We all know that the heat shock proteins, they actually respond to heat stress, and they play a crucial role in the stress response pathway. So the next question comes into my mind, that are heat shock proteins expressed? And if the heat shock proteins expressed, how they respond to elevated temperature? So when we started this study, we have seen that there are several roadblocks. The first, but though we are very encouraged after seeing that yes, there are some previous publications which have indicated that heat shock protein 70 is present in this organism, and the same study actually showed the expression of HSP70 protein, which actually has been shown to be increased with increasing temperature. So we had a long chat with Shumit and Shumit's lab, and we plan to check the expression of the other heat shock proteins, the HSP60 and HSP90, as a function of temperature. So when we are talking about checking the expression, we are actually interested to see that how the RNA of this particular gene has been changing as a function of temperature. But before just starting that particular experiment, we noticed that there is no sequence information available for those HSP proteins. And after some literature search, we found that Bellamia originated in Africa and dispersed to Asia. But interestingly, it is diverse from the African population due to strong geographic isolation. And this is really interesting to me because it has been shown that only not the genetic composition is important, but the environmental factors or that particular geographical location is changing the behavior of that or the genetic composition of that particular species. So what we actually have done, or we wanted to try, that we identified, because we could not find the sequence information for that particular mollusca, we identified two other family members, Pomicea and Biofamiliaria. So these are other two similar types of gastropods, and in these two different types of gastropods, the sequences of the three proteins HSP60, HSP70, and HSP90 are known. So we analyzed the protein sequences and we analyzed the cDNA sequences of these two species. And we surprisingly found that though these heat shock proteins are very well known for its evolutionary conservation among the higher eukaryotes, but we found that these proteins shared like less than 30% sequence homology. So now if you can understand that when the two proteins can share less than 30% sequence homology, then probably their cDNAs also diverge a lot. And we noticed that the cDNA sequences also diverge a lot. But we need to design the primers. So we need to focus on the cDNA sequence. So whatever the small stretches of sequence conservation we found when we aligned the sequences from these two species, Pomicea and Biofamiliaria, we designed the primers. And when we tried to do a PCR amplification of this HSP 60, 70, and 90 from Bellamia, we could not get any positive hit. So the primers could not detect any gene encoding those proteins in Bellamia. So what could be the reason? The reason probably is like the species, though they belong to the same class, they have a divergent set of sequences. So whenever we design the primers, that could not pick up HSP 60, 70, and 90 from Bellamia species. So then we thought we will try a next generation sequencer, and then we would be able to determine the relationship of these particular proteins, like HSP proteins, with elevation in temperature. And not only that, probably we would also able to see with that next generation sequencing approach, like all other different genes and their expression with the elevation in the temperature. So the future would lie for marine ecology. I think that this is the, like, from the molecular biology perspective, that the high throughput sequencing and transcriptomic analysis. Because I think that's the right time to use or to look at the ecological approach with that much detail, so that we would be able to know, like, what are the players are playing or controlling the different responses at the molecular level. And this would also help us to understand the basis of the divergence between the species. But we need to be careful about the proper sampling, because a particular geographical location, already there are several reports, like the geographical location matters a lot when you are like conducting the study of expression studies of this microbial organism with respect to its habitat. But if we can carefully consider those things, the blending of this molecular aspect with ecological problems will greatly impact all the different applications of marine ecology. And metagenomics, with its powerful tool and its advancing every day, will help us to understand it in a greater detail. So with this, I would like to thank all of you again, because you were like hearing a talk in a Sunday afternoon. And for the students, I'd like to share, it's a very beautiful statement from one of the great leaders. I like it very much. So this is for about the new generation like you. So think in a broader way. Don't just limit yourself that you are like studying a particular stream. Remember, this is a new era, and after this COVID, again, we all will need a new skill sectors, and science is now interdisciplinary. So don't limit yourself just to know about only about ecology or molecular biology or microbiology. Try to think in a broader way. So with this now, I'd like to end here, and thank you all. Thank you, Shumit, for giving me the opportunity to talk in this platform, and I'd be happy to take any question.
Thank you, Chandrama, for a nice presentation. And the participant must be very much happy with this presentation, because I think you have touched all the aspects of what are the molecular biology application in marine ecology. So the participant who don't know about Chandrama, due to the introductory slide, might be you were not there that time. So Chandrama basically is not a marine person or the aquatic person, but I'm very glad that I found a colleague like her, and whenever I have any discussion, and I am not so very much and she also highlighted some part of this. So we are very much in still it is infancy till the future about our research, but we are trying to do something for the Sundarban estuarine ecosystem, marine biology, and with the application of molecular tools in those areas. So thank you again, Chandrama, for your nice and explanatory presentation. I really enjoyed it basically. So you covered almost all aspects what can be done in the molecular biology or the application of molecular biology in the marine or aquatic ecosystem. So vote of thanks and take some questions from the participants. Over to you, Priya.
Yeah.
Thank you, sir. Thank you for giving me the opportunity to propose a vote of thanks. On behalf of Marine Ecology Laboratory and the entire Aquafile team, we would like to acknowledge our gratitude to you, ma'am, for your magnificent and informative speech about the molecular biology tools to study aquatic ecosystem. And I mentioned our deep sense of appreciation for your explanation how molecular tools can be effectively be used in the field of marine science. And as we all know, Our government has given much importance in blue economy. So we strongly feel there are much opportunities for young researchers in the upcoming decades um, for in the field of marine science, and ma'am has touched almost all the aspect of molecular biology and how we can use it in the field of marine ecology. So before starting the Q&A session, I want to thank all the participants for your active participation and asking us a pertinent questions. So there are a number of questions, but due to limited time, we can't take up all the questions. There are a few more questions I want to discuss with ma'am. The first question is from Chanchal Kumar De. He asked how molecular biology affects on aquatic diversity. So
Uh,
now
so,
over to ma'am.
okay, so this is the question from Chanchal. Chanchal, I don't think the question is, like has rightly asked, because molecular biology is a technique. It cannot do anything by itself. If you want to ask some question, then you can take the help of the molecular biology tools. It's not that molecular biology is doing something to aquatic ecosystem. No. If my message gives you that, then I'm sorry. But this is great. This is a very strong, powerful tools, the molecular biology techniques what we have, and that I have discussed some of it, not all of it I have discussed today. But with those techniques, for example, with the genomics, with the expression studies, you can actually determine the relationship of a microorganism which is present in the aquatic ecosystem with its habitat. It's only that. Like we can use this technique to understand aquatic ecosystem in a greater detail. Did you understand? So
I think
yeah, it's not like something like molecular biology is doing something to aquatic ecosystem. No, because it's a very wrong conception among the students that actually we also face when during our class. This is just a technique. So molecular biology means this comprises many techniques, and in biology there can be a lot of different questions, and how you are going to address those questions. So that time you can take help from either from the molecular biology techniques or from the microbiological techniques or any biochemical techniques. So those are the techniques you actually are going to use to solve your question.
okay, ma'am. I think he got his answer. The next question is from Sakura Konar. She asked, can determining of microbial species diversity help in evolutionary studies?
Oh, sure. Yeah. If you just determine the microbial diversity, it's definitely help you to understand the evolutionary studies. Because if you remember, like what I have shown in my presentation, with the help of this 16S rRNA technique, you can actually would be able to draw the phylogenetic relationship. And that phylogenetic relationship will help you to determine the evolutionary relationship.
Okay. The next question is from Orja Chakraborty. Ma'am, shotgun sequencing is also a molecular method. Can you please tell us about its advantages and disadvantages?
Shotgun technique is a powerful technique in that sense that with this technique, you can cover a lot of sequence. Suppose you have really, if you want to use it in a metagenomic study where you can actually isolate like more than 100 microorganisms, and then you can use this technique to determine what are the different species are present. And the disadvantage of this technique, that usually this technique involves a like lot of computational analysis, because if you remember that I have shown that usually first you need to like fragment the genomes and then it will give you the sequences, but all the sequences are like not properly aligned. So all those are very randomly organized, and the different tools help us to align or to match those sequences. So suppose if there is any glitch in that aligning software, then probably you will get a wrong, you will actually have a wrong information on about the sequence of that particular microorganism. So you need to like do some different complementary studies. You just can't only rely on one experiment and come with a conclusion.
Okay, thank you, ma'am. This is the last question from the Q&A session. Kia Sarkar has asked, what is the difference between whole genome sequencing and next generation sequencing?
Whole genome sequencing is where you are actually sequencing the genome of a microorganism. And next-gen sequencing is completely different. Next-gen sequencing is for mainly the expression studies. And here you don't need to clone anything, clone any gene. You can just isolate the RNA or you can isolate the genomic DNA, and then you can do a sequence analysis. So these two are completely different, okay? Genomic analysis means where you need to clone the genome and then you can analyze by sequencing. But from the genomic sequencing, you cannot do an expression study. And it has to be only DNA. But the NGS actually employs both DNA and RNA. And this is, you will get the genetic information, like the sequence information, and also you will be able to know the relationship of that particular organism with its habitat. So suppose you want to work with a acting gene, and you want to determine that with some different conditions in the environment, how the acting gene is changing its expression. So that answer you only get from the NGS study, but not from your genomic study. I hope that it will clear your confusion.
Hello, Chandrama. Can you
So.
please stop presenting this slide? Then your face will be visible.
Yes, ma'am.
Yes, sure. So can you see it?
No, ma'am.
No, you go, stop presenting.
Presenting.
Okay, just give me 2 minutes.
Yes, perfect.
So, ma'am, can we share your email ID with the participants for their future queries?
Yeah, sure. You can share my email ID. So now can
Okay,
you
thank
see?
you, ma'am.
I don't know. Probably I have some problem with now the connectivity.
No, it's okay. Your face is visible.
It's okay, ma'am.
Anyway, thank you, Chandrama. And do you have any questions here? Is there any other question?
No, ma'am.
Yeah,
Okay.
if
No
you
other
have any
questions.
question, you can always contact me through email only.
Yeah. So the
Okay.
participants can contact with Chandrama with our email address.
For
So
their future
anyway,
queries, okay.
we have just come to the end of this session. And once again, Chandrama, for your nice presentation, and moreover, you have taken out time from your busy schedule. I know how busy you are now these days. So anyway, very much thankful, and I'm really humbled and overwhelmed that you readily accepted my invitation. Even though you are not an aquatic biologist, but you tried your level best to give the presentation in a perfect fashion.
Yeah, for the first time in my life, I actually go through some particular definitions of this.
Yeah. So I know how difficult it is, but anyway, our future collaboration will more enlighten both of us, and will be more educated. It will be complementary for each other. Anyway, thank you, Chandrama, for your nice presentation and time. Thank you.
Thank you. Bye.
So the next talk will be at 4 p.m. Indian time by Dr. Gabriel Plavan from Romania. Till then, stay tuned. See you at 4 p.m. Thank you.